The overall goal of this procedure is to localize low-level expressed odorant receptor genes as well as other genes in the insect antennae using digoxigenin-labeled or biotin-labeled probes.
This method can help answer key questions in the neuroscience field, such as the identification of putative genes and the localization of target genes in tissue or cells. The main advantage of this technique is that it can let us to visualize and localize the target genes at the cellular level.
We first had the idea for this method when we wanted to localize some memory proteins which were difficult by immunocytochemistry localization experiments.
To begin this procedure, select the new molting adult locusts that are active and have intact antennae. Then, cut the antennae into 2 to 3 millimeter pieces using sterile razors. Next, apply OCT compound on a freezing microtome holder and place the samples on the compound horizontally. Transfer the holder into the freezing microtome at -24 degrees Celsius to equilibrate until the compound freezes. Afterward, take out the holder and cover the samples with a little compound. Transfer the holder into the freezing microtome at -24 degrees Celsius again for at least 10 minutes. Then, section the frozen samples into 12 micrometer thick slices. Subsequently, thaw mount the slices one by one on slides and air dry them for 10 minutes.